Hello students, today's video is a complete comprehensive overview of insulin. As we all know, insulin is a hormone produced by the pancreas. In today's video, we will first study exocrine and endocrine pancreas. After this, we will study structure of insulin, process by which insulin is synthesized by pancreatic beta cell. Once synthesized, how it is released from beta cells in the blood, then mechanism by which insulin binds to its receptors on the target cells and produces its action. We will also discuss functions of insulin, how insulin regulates blood glucose levels and is essential for carbohydrates, proteins and fat metabolism. Now look at this figure. It shows structure of pancreas. Pancreas is a long, slender, thin organ around 12 to 15 centimeter long. It is located in the abdominal cavity. This part of the pancreas is called as the head. It lies in the curve of duodenum. Now very important to remember, pancreas is an endocrine as well as exocrine gland. Exocrine pancreas consists of large number of lobules and each lobule is made up of acinar cells. These cells produce pancreatic juice shown in green. This pancreatic juice is released locally in the duodenum. Endocrine pancreas is made up of pancreatic islets or islets of Langerhans, which produce hormones and release them directly in the blood. Now, this is the section of pancreas composed of pancreatic acini and this is pancreatic islet or islet of Langerhans. Now, each pancreatic islet is composed of four types of cells: alpha cells, beta cells, delta cells and PP cells. All these cells produce different types of hormones and these hormones are released directly in the blood. Approximately 75% of these pancreatic islet cells are beta cells. Beta cells produces insulin. Insulin is essential for the transportation of glucose from the blood to the body cells. When the glucose is taken up by the body cells, levels of glucose in the blood reduce. Now, approximately 20% of these islet cells are the alpha cells. Alpha cells release hormone that is called as glucagon. Glucagon increases the levels of glucose in the blood. Now, 4% of these pancreatic islet cells are the delta cells. Now, delta cells produces a hormone called as somatostatin. Somatostatin inhibits release of insulin as well as glucagon. Now, approximately 1% of cells are pancreatic polypeptide, that is PP cells. Now, these cells regulate exocrine and endocrine secretion as well as appetite. So, insulin is an anabolic peptide hormone produced by beta cells of pancreatic islets, also called as islets of Langerhans. Anabolic means constructive. Now, insulin is known as anabolic hormone as it promotes synthesis of glycogen, proteins and fats. Now, as we all know, when we eat food, carbohydrates in the food are broken down to glucose and the glucose is absorbed in the blood. This increases levels of glucose in the blood. Now, look at this figure. This is a cell. This is insulin receptor and this is a glucose channel which is closed. Now, as soon as this insulin binds to insulin receptor, the glucose channel open and now glucose from the blood is transported inside the cell. So, insulin is essential for the uptake of glucose by the body cells. And as this glucose moves inside the cell, level of glucose in the blood falls. So, insulin reduces the level of glucose in the blood. Now, in the absence of insulin, this glucose channel remains closed and the glucose cannot move inside the cell. However, there are few exceptions like brain cells, liver cells, RBCs, kidneys do not require insulin for the uptake of glucose. So, the most important function of insulin is to facilitate uptake of glucose from the blood into the body cells. And this glucose is utilized by the cells to produce energy. Insulin also promotes storage of energy in the form of glycogen, proteins, and fats. Now, the glucose that is not utilized by the cells for energy is converted to glycogen and is stored in the liver and muscles. Insulin also promotes synthesis of proteins from amino acids, and these proteins are stored in the muscles. Fats are synthesized from fatty acids, and these fats are stored in the adipose tissue. Now, glycogen, proteins, and fats are stored and they are utilized by the cells for energy whenever the levels of glucose fall in the blood. Thus, insulin is a master regulator of carbohydrates, lipids, and protein metabolism. Now, let's study structure of insulin and how insulin is synthesized. By now, it's clear that insulin is secreted by pancreatic beta cells. The gene that codes for the synthesis of insulin is the INS gene, and it is located on the chromosome 11. Now, information carried on this gene is used to synthesize the peptide hormone, that is insulin. Now, rough endoplasmic reticulum. Ribosomes of rough endoplasmic reticulum produce pre-proinsulin. Now, this pre-proinsulin is made up of 110 amino acids. Now, this pre-proinsulin has 4 segments: signal polypeptide, B chain, C chain, and A chain. Now, when this pre-proinsulin is transported from endoplasmic reticulum to Golgi apparatus, signal polypeptide is cut or cleaved off. The remaining structure is called as proinsulin. Now, Golgi apparatus packs proinsulin into secretory vesicles, and these secretory vesicles are found to be present in beta cells. Now, in secretory vesicles, this proinsulin is stored as insulin and C-peptide. So, this is the structure of insulin. As insulin is a peptide hormone, it is made up of amino acids. So, insulin is made up of 2 chains. This is the red color A chain, the A chain that is shown here in the red color, and B chain that is shown here in the blue color. Now, A chain has 21 amino acids while B chain has 30 amino acids. Now, A and B chains are linked by 2 disulfide bonds. In addition, this A chain has 1 intramolecular disulfide bond. Now, when beta cell is depolarized, insulin and C-peptide are released together from the secretory vesicles stored in the beta cell. Now, let's see how insulin is released in the blood when beta cells are depolarized. Now, let's understand mechanism by which insulin is secreted or released from the pancreatic beta cells. Insulin is found stored in these secretory vesicles. Now, low or basal level of insulin is consistently secreted from the beta cells. But as soon as the blood glucose rises, that is after a meal, secretion of insulin increases. So, stimulus for secretion of insulin is the level of glucose in the blood. Thus, glucose stimulates insulin release from the beta cells. So, look at this figure. It depicts the structure of a pancreatic beta cell. Now, GLUT2 are the glucose transporter 2. Now, these GLUT2 are the channels that permit influx of glucose inside the beta cells. So, GLUT2 transport glucose from the blood into the beta cells. Now, transportation of this glucose is in proportion to the concentration of glucose in the blood. More glucose in the blood causes more influx of glucose in the beta cells and therefore more release of the insulin, while less glucose in the blood causes less influx of the glucose and therefore less release of the insulin. Now, once this glucose is inside the beta cell, glucose is phosphorylated to glucose 6-phosphate by the enzyme glucokinase. Now, this glucose 6-phosphate enters glycolysis pathway and pyruvate is formed. Now, this pyruvate enters citric acid cycle or the Krebs cycle and also undergoes oxidative phosphorylation in the mitochondria. Now, this produces adenosine triphosphate, that is the ATP. Now, these ATP inhibit ATP sensitive potassium channels and thus ATP sensitive potassium channels close. So, now potassium does not move out of the cell and thus concentration of potassium increases inside the beta cells. Now, this increases membrane potential and further causes depolarization of the cell membrane, opening the voltage gated calcium channels. Now, as these voltage gated calcium channels open, there is influx of calcium inside the cell. So, concentration of calcium increases inside the cell. Now, increased calcium causes exocytosis of insulin. So, these vesicles containing insulin, they fuse with the cell membrane and insulin is released in the blood. So, this is how levels of glucose in the blood regulate secretion of insulin from the beta cells. Now, other factors, for example, incretins such as GLP-1, that is glucagon-like peptide, then GIP, that is glucose-dependent insulinotropic polypeptide, bind to their G-protein coupled receptors on the beta cells and also promote exocytosis of insulin. So, this is how insulin is released in the blood from the beta cells. Now, once insulin is released, let's understand how it binds to its receptors and produce physiological functions. So, let's first discuss the mechanism of insulin action. This slide explains mechanism of insulin action. Now, look at this figure. This figure shows a cell. This is a cell and this is a plasma membrane. Now, to produce its effect on the target cell, insulin binds to the insulin receptors and activate them. This is an insulin receptor. Now, insulin receptor is made up of 4 subunits, 2 alpha subunits and 2 beta subunits. Now, these subunits are held together by disulfide linkages. Now, insulin receptor is an enzyme-linked receptor. Enzyme tyrosine kinase is found attached to the beta subunit. So, insulin receptor is a tyrosine kinase receptor, and it is made up of two extracellular alpha subunits and two transmembrane beta subunits. Now, as shown in the figure, these alpha subunits lie entirely outside the cell membrane, while the two beta subunits pass through the cell membrane. Now, two insulin bind to these two alpha subunits. Now, binding of insulin to insulin receptor causes autophosphorylation of enzyme tyrosine kinase. On phosphorylation, tyrosine kinase enzyme gets activated and cause phosphorylation of IRS, that is insulin receptor substrate protein gets phosphorylated. Now, on phosphorylation, this IRS protein becomes active and in turn activates other proteins. So, a cascade of phosphorylation and dephosphorylation comes into motion. Now, this causes activation or inhibition of enzymes responsible for producing metabolic functions of insulin. Now, insulin transports GLUT4, that is glucose transporter 4, from the cytoplasm to the cell membrane. Now, this increases transportation of glucose from the blood to the muscle and adipose tissue. Now, insulin also facilitates synthesis of glycogen from the glucose. This glycogen is stored in the liver and muscles. Insulin also mediates synthesis of fats and protein. Now, glycogen, fats and proteins are the storage form of energy. So, now let's discuss metabolic functions of insulin in detail. Now, this is a slide that summarizes effect of insulin on carbohydrate, fat, protein, metabolism in the liver, adipose tissue and muscles. Now, before discussing metabolic functions of insulin, let's understand few terminologies. First is the glycolysis. Glycolysis is a process where glucose is utilized by the cells to produce energy. Then glycogenolysis. Lysis means broken down. So, glycogenolysis is the process where glycogen is broken down to produce glucose. Then gluconeogenesis. Normally, glucose is released when the glycogen break. But gluconeogenesis is the synthesis or it is a production of glucose from non-carbohydrate sources like amino acids, fatty acids. Then lipogenesis, that is the synthesis of fats. Lipolysis, that is the breakdown of fats. Let's first understand carbohydrate metabolism. Now, as we all know, immediately after a meal, blood glucose level rises and glucose stimulates increased release of insulin from the pancreas. Now, insulin mediates rapid uptake of glucose by the liver cells, by the adipose tissue and also by the muscles. So, uptake of the glucose by the liver cells increase. Now, glycolysis increases and this glucose is utilized by the cells to produce energy. Excess glucose not utilized for the production of energy is stored immediately in the liver as glycogen. So, synthesis and storage of glycogen increases. Now, insulin inhibits the process of glycogenolysis. So, glycogen is not broken down. That means glycogen is not converted to glucose. Insulin also inhibits the process of gluconeogenesis. So, glucose is also not synthesized from the non-carbohydrate sources. So overall effect of insulin is to increase utilization of glucose by the liver cells and to reduce synthesis of glucose. So this reduces blood glucose levels, and glucose is primarily stored as glycogen in the liver. Now, insulin also stimulates process of lipogenesis. There is increased synthesis of triglycerides. Now, excess glucose that is not converted to glycogen is now converted to fatty acids. Now, these fatty acids are converted into triglycerides, and these triglycerides are packed as very low density lipids. That is, VLDL are synthesized by the liver. So, these VLDL that are synthesized by the liver are transported via the blood to the adipose tissue. And here in the adipose tissue, these triglycerides are stored as fat. Now, in addition to this, insulin also inhibits the process of lipolysis. That is, insulin inhibits breakdown of lipids. So, overall effect of insulin is to promote the storage of fats in the adipose tissue. Insulin also inhibits breakdown of proteins in the liver. So, insulin increases uptake of glucose in the liver. It increases synthesis of glycogen. It mediates storage of fats in the adipose tissue and also prevent breakdown of proteins. After liver adipose tissue, insulin stimulates uptake of glucose by the adipose tissue. Thus, glucose is mainly utilized for the synthesis of glycerol. Now, as already discussed, VLDL synthesized by the liver is transported to the adipose tissue. Now, main component of this VLDL is the triglyceride. Now, enzyme lipoprotein lipase present in the adipose tissue splits or break triglycerides in the VLDL to fatty acids. Now, these fatty acids combine with the glycerol and produce the fat. Now, this fat is stored in the adipose tissue. So, insulin causes increased fat synthesis and storage of fats in the adipose tissue. Insulin also inhibits process of lipolysis and thus it prevents breakdown of fats. Now, effect of insulin on muscles. Now, insulin stimulates uptake of glucose by muscles. Glycolysis increases. So, glucose is utilized to produce energy, that is to produce ATP. Now, excess glucose not utilized to produce energy is converted to glycogen and it is stored in the muscles. Now, insulin also stimulates increased uptake of amino acids from the blood into the cells of muscles. Now, these amino acids are utilized for the synthesis of proteins. Insulin also inhibits protein breakdown. Thus, insulin is an anabolic hormone as it promotes synthesis of glycogen, synthesis of fats and synthesis of proteins. So, this is a complete comprehensive overview of insulin. If you find the video useful, kindly like, subscribe and share this video. Thanks for watching this video.